introduction about who I am. So I'm quite a junior researcher compared to all the amazing stellar researchers that you've heard so far in the series. So I've just completed my my PhD in marine science at the University of Western Australia, and I am what would be typically called a coral reef scientist because I study coral reefs across various levels and scales, from having studied the coral holobiont, or how corals behave on more physiological levels under stress conditions, but also now from my current research, which I'll actually just show a bit about at the start of the presentation, about how I'm using landscape level coral reefs to understand how they interact to build these beautiful large scale landforms that we call reef islands. So very diverse multidisciplinary skill set that I have. And this is kind of developed over the last 10 years of my research career thus far. So I'm going to give you a bit of a brief background as to where I've come from, particularly for those students who want to pursue marine science going forward into the future. So a bit about my current diverse backgrounds, like I mentioned. I'm a PhD candidate. I also have a really important role in the communications sector of both the Oceans Institute at the University of Western Australia and, of course, with the Western Australian Marine Science Institution, which I'm very proud to represent. So my expertise currently is focused in the area of coral reef and coral reef island geosciences, but I also have a diverse scope and background in spatial science, so using GIS and remote sensing, coral reef ecology, and of course, coral ecophysiology and the holobiont response, which I'll talk about in detail in this presentation. So I know most of you might be wondering, how do I go about actually pinpointing what degree I should actually undertake when you step out of high school into what we call the real world and doing a real world degree that's going to be applied? So I didn't do an undergraduate degree in marine science or marine biology. I actually came from a very diverse background, studying more of terrestrial and freshwater conservation ecology. So I conducted my Bachelor of Science, and it was a Bachelor of Science with a major in wildlife and conservation biology at La Trobe University in Melbourne. And this was back in 2011. That feels like an absolute, you know, time ago. It feels like yesterday at some point, but it just shows how much your career can evolve over the course of your degree. So this was an awesome degree, and I'm so glad that I actually chose to do it because it gave me a very diverse skill set. And it actually allowed me to co-major across 3 different disciplines. So I was quite adamant that I might become a plant ecologist or a botanist at one stage. So I actually did a sub-major or co-major in botany, ecological and evolutionary genetics, and also zoology. And here's just a diverse example of some of the amazing field trips that were on offer, from arid zone ecology, alpine ecology in the Great Alpine National Park of Victoria, and conservation ecology down at Wilson's Prom in Victoria. I only branched into marine science into my 3rd year, where I took 2 really pivotal subjects, which was marine physiology and coastal marine ecology. And this kind of set me up with this multidisciplinary degree across both terrestrial and marine systems, which I could then take further into my higher education. So my first real taste of what I wanted to pursue in science, in particular in the area of marine science, was actually on the 3rd year field course I did that I did with La Trobe University in marine physiology and coral reef ecology. And this was actually up at the amazing Heron Island research station on the Great Barrier Reef. Now, this is an amazing study site because you can actually have a natural field laboratory right off these islands. And it was such a unique experience to do a 3rd year subject in such a beautiful and stunning location. And it was here that I actually was motivated to pursue a further degree in marine science. I fell in love with coral reefs, particularly being able to do all these incredibly amazing and diverse experiments here at the Heron Island Research Station. Everything from testing the physiology of crustaceans to echinoderms, as well as looking at how the intertidal migration of fish actually dictates how different species of fish move in and out of these reef systems during the high and low tides, and what type of physiology controls this intertidal migration of species. So it wasn't until I got, I'm going to jump now 10 years later to kind of give you an introduction of just what I am currently working on in a quick nutshell, and then I'll backtrack again to talk about my honours degree that actually came after my bachelor. So just very quickly, like I said earlier, I study these amazing dynamic landforms called reef islands. These are actually coral-built landforms. Now, all that coral reef environment, the productivity of all its different organisms, corals, mollusks, forams, over time, over geological time periods, actually help to build these landforms. Now, I'm talking on the order of 6,000 to 7,000 years of reef history. Now, by understanding how these reefs actually function, we can actually better predict how islands grow and may erode with climate change. And we might be thinking, well, why are these landforms so critically important? Well, they actually play these pivotal habitats for many of our most threatened and vulnerable species. They're our rookeries for endangered green, hawksbill, and flatback turtles, but they also provide critical roosting grounds for many of our sea and shorebird populations as well. So understanding the function of the reef and how it produces these sands or sediments is going to be pivotal at predicting how these islands might change under projected climate change scenarios. So now when I talk about sands and sediments to coral reef islands, I'm talking about the building blocks that actually build the landform. Now, these are not islands like Rottnest. These are actually purely built by the biology. And that's quite an extraordinary feat that you have a living reef system that's actively producing sand, or we call sediments, in geoscience, that actually breaks down over very long time frames and then gets kind of channeled on top of the living reef to start building this landform over those 6,000 to 7,000 years of reef history. Now, when I talk about the building blocks, I'm talking about everything from those corals that actually build the reef framework, so those hard and soft corals which might be breaking down naturally, as well as your gastropods and bivalves which form the phylum Mollusca. All that shell debris breaks down over time, produces sands, as well as even single cellular protists like these foraminifera that you see just next to the mollusk and gastropod images. These can be microscopic to about the size of a 20-cent coin, and they actually are pivotal in building many of the world's reef islands. It's not just animals that build islands, it's also plants as well. So we have Halimeda, which form very beautiful biohomes, so large extents of meadows. And this plant actually produces the mineral calcium carbonate, just like corals, just like mollusks do. And over time, those minerals actually accumulate in the system and help to build these landforms. So pretty spectacular landforms. And my current role is trying to understand how the reef is actually producing the material over these long-term timeframes and better predict how this material might change into the future. Say we have a mass bleaching event which wipes out extensive parts of a reef system adjacent to these islands. So this is a really real-world application of how you can combine different fields from geology and ecology to understand more holistically on a broad scale how to better predict and conserve these systems. So let's backtrack and just get a bit of an intro as to why coral reefs are critically important and what exactly are these structures. So I'm sure this has been already addressed in great detail in your classes, so I won't go into overly too much detail, but on a large scale perspective, coral reefs are these three-dimensional structures. Now we can see them from outer space. They are composed primarily of the skeletons of what we call hard corals or hermotypic corals. These are typically the fast-building corals on the reef, like our Acropora or Bacillopora species. And these, over time, over long time frames in the order of several thousands to tens of thousands of years, actually build and allow the reef to grow vertically towards the top of the water. And most reefs over these long time frames are actively building due to this natural life and death cycle of these corals and other organisms. So corals, what exactly are these foundational species? Well, they're a symbiotic kind of a composite organism, I'm going to call it. These are of the family Cnidaria. So they belong to the same family as our anemones and jellyfish. They're a polyp, so a small single cellular organism which actually forms a symbiotic relationship with a single-celled algae called Zooxanthellae of the phylum Symbiodinium. Now, this is a really critical relationship because not only does the coral host provide shelter for the alga, it is actually working in complete partnership with it, providing nutrients both to the alga, and then in turn, the algae is actually producing enough glucose, sugars, and other growth important compounds, which allows the actual animal to grow and build its skeleton, that calcium carbonate skeleton, of which builds the broader foundation of the reef. So a very critical evolutionary relationship. Now, we know that coral reefs, they play critical importance in supporting large amounts of reef biodiversity, supporting 25% of all known marine species, including many of our rookeries and nursery grounds for many of our juvenile fishes, which are important for recreational and aquacultural purposes, as well as the larger megafauna that rely on these reefs as well. So in terms of their economic benefit, they are critically important, providing $30 billion in benefits to many of the world's coastal nations, from the Indo-Pacific region to the Caribbean in particular. That's providing food, fisheries, and tourism support as well. But we are fully aware of the current threats to coral reefs into the 21st century, and these range from everything from acidification of the ocean, increasing SSTs or sea surface temperatures, water pollution based on land runoff from agriculture and other pollution sources, and of course, overexerted fishing practices on these reefs as well. So these composite organisms, corals, they come in diverse shapes, and that actually enables them to behave in very unique ways on these reef systems. So we have very different morphologies of corals on these reefs. We have our fast-growing corals. So these include our tabular and plating corals, our branching corals that are some of the most common and fast-growing species, and our corymbros corals as well. These are our faster-growing. They grow over very fast time frames and are actually the major reef builders, those hard or hermatitic corals. Now, when it comes to the other groups, such as encrusting and mound-like corals, these are generally much more sturdier. They grow over much more slower time frames, and importantly, it's these types of corals that are actually more resilient to disturbance. It's these other three morphotypes, as I'm going to call it here, our branching, tabular, and corymbros, that are actually more susceptible to acute disturbance event like SSTs, so increasing temperatures, cyclones, cyclone damage, and other disturbances, either from competition or predation. So I'm going to hint back at these key ideas as I move into the focal point of the presentation. So just from now on going forward, just remember that it's these three critical morphotypes that are actually the most sensitive to climate change. And it's our encrusting and mound-like corals, which are going to be able to future-proof our reefs into the 21st century. So now I'm going to talk about a case study which I actually conducted during 2014 to 2015 back at Heron Island on the GBR. Now, this was actually formed part of my Bachelor of Science honours that was conducted at James Cook University in Townsville, once again, an eon ago. So what I wanted to do here on Heron Island is I wanted to actually sample these more fragile coral morphotypes, in particular the branching morphotypes. And I wanted to understand how they were actually going to recover under a combination of both high temperatures. So these were going to be temperatures that were just really close to what we call the bleaching threshold, the point just before corals bleach, but also in combination with how they actually cope with injury. Now, an aftermath of high temperatures on reefs is actually increased cyclone severity and damage to these corals. I wanted to look at how these combination of effects were going to impact the future recovery of these very fragile species. So we need to understand some basic facts first. So under high water temperatures, we have an acute disturbance called coral bleaching. Now, this process actually occurs when you have a higher than abnormal duration of warm temperatures that hangs around on a reef for more than a week or 2 weeks. Now, what this actually does, it actually prompts the coral itself to actually exert a stress response. Now, under high temperatures, the alga actually starts to produce a toxin called a reactive oxygen species. Now, what the coral host does, it actually recognizes this toxin and then detects it as at being at too harmfully high levels. Now, just like any organism, it's going to reject these alga if the toxins become in too high concentration in the tissue. And that's exactly what the bleaching response is. The coral animal detects these toxins. It then responds by expelling the zooxanthellae from its tissue, resulting in this translucent appearance that we see as the coral bleaching response. Now, particularly, this is acute in these high susceptible corals, our branching, coriombros, and plating corals. And that's because they have a very limited tolerance range to these very high temperatures. In the case of bleaching, this typically occurs around 34 degrees Celsius, when the normal reef temperatures are actually hanging around about 24 to 26 degrees. That's significantly hotter. Another aftermath of these hot temperatures is actually an increase in storm surge events and cyclone severity. These become more common under climate change induced temperatures. So you tend to have a transition from these very complex reef structures to these rubble fields of highly degraded reef structures. Now, as you can see in this example here, most of the plating fragile corals and branching that you see in the image on the left have actually been completely decimated under this event. So once again, it shows that these morphotypes are going to be increasingly susceptible to these disturbance events going forward into the future. So we call this process coral fragmentation, and that typically happens to these branching types. Now, it's those much more resilient morphotypes, our mound corals. In this case, you can see the massive coral parietes here that are going to actually be the saving grace of these reefs. They're very resilient to bleaching and very high energy event disturbance, and therefore will kind of be the remnants of these reefs going forward. And I actually recently just saw evidence of this in the Maldives this actually last week, where they've had a consecutive amount of high energy events and bleaching over the last several years. And you have a lot of these mound corals left, which are actually producing or maintaining the rest of the reef, the living reef framework. And it's quite sad to see over quite short-term durations. Now, so how do we mimic this? So we actually wanted to replicate this under controlled conditions at Heron Island. So we actually ran a 12-day experiment to monitor the coral stress response and growth response under both high temperatures and injury. And this is to mimic these climate change impacts that I was just talking to you about. So what we did here is actually, these are 3 of my colleagues, 2 from Portugal and 1 from JCU. And we actually went out on a beautiful day at low tide and started sampling these intertidal reefs. This is the reef flap of Heron Island. And we sampled a very particular species called Acropora aspera. Now, Acropora aspera is a highly robust case study species. It's been studied quite regularly in the past, and therefore it's a very good case study to use in any controlled experimental conditions. Now, once we collect these corals off the reef flats, we need to let them acclimate. And when I say acclimate, they need to start to stabilize and become less stressed. So if you think you're being broken off from your colony, you're going to exert a lot of stress. These corals are going to be highly stressed out, and they're going to be out of their comfort zones. They need to be able to relax a bit over time. So we transported them about 100 meters from their home on the reef flat, put them into these holding tanks, which can hold at least 1,000 liters of water, which get to pass through every few minutes. It's quite a lot of water recycling through these systems. And these corals sat in these tanks for about a week, and that allows them to get used to their new conditions. So as you can see, we've got a couple of fragments in these tanks, and we had replicate tanks that we're going to then simulate both of these important conditions that I'm going to talk about. So we then placed them into 2 consecutive tanks. Now, one of these tanks was actually held at the normal reef temperature, which was about 24 to 26 degrees on Heron Island during March of that year, which is quite stable temperature for the Southern Great Barrier Reef. Now, the second tank, we then wanted to mimic climate change predicted temperatures. So we actually ramped the tank's temperature to about 32 degrees. Now, noting from the previous slides, this is not the bleaching temperature. This is just below the temperature at which the corals would start to exert a bleaching response. So they're going to be stressed, but they're not going to bleach. And then by doing so, we could actually then replicate these high temperatures that would simulate these increased sea surface temperature events on the Great Barrier Reef. Now, to replicate injury after a cyclone event, half of the colonies in each of these tanks actually had the tips removed from the top of the colony. So we actually got a pair of secateurs and cut the tips off the top of these corals. And that was going to simulate a cyclone event ripping through this reef and fragmenting the coral branches over time. So there we go. We have an experiment where we can simulate both temperature stress and injury stress under climate change predicted scenarios. So once again, this is to notice that we actually divided the tank in half. Half were injured, half were left intact with their tips. And that's going to help us to ensure that we have control corals that we can then compare the results to. So how exactly do we measure the stress response of these corals? So let's see what happened under normal reef temperatures. So we actually photographed the corals under 3 different microscopy fluorescent regimes. We did what's known as just general light microscopy. We did what's known as GFP, which is the green fluorescent protein, and then just under a scanning electron microscope to look at how the skeleton grew back. Now, if we note at the far end of the panel at day 2, that's the start of the experiment where the tips had just been cut off. So we expect no regrowth to happen here. Now, if you noticed, after we sampled every 2 days, the coral tip that we had cut actually grows back to full extension by day 12. That's a really rapid growth rate recorded for this species. Now, what's really important here when we talk about the second panel for green fluorescent protein, this protein, this green protein that I've actually photographed, plays a very important role in protecting the top of the coral from any damage from UV radiation and high temperatures. So as you can see, as we move towards day 12, the coral starts to produce this protein in very high amounts. That actually allows the tissue and the skeleton to better grow and function without being damaged by other disturbance processes. So the GFP is actually acting as a protective mechanism, preventing any further damage and therefore aiding the tip to recover over 12 days. And of course, we see that exactly the same in the SEM images, where the calcification of the skeleton actually regenerates alongside the tissue. So we have, under normal reef conditions, perfectly healthy colonies. They might have been damaged, but they've been able to rejuvenate themselves and recover to their full extent of growth by the end of the 12 days. So pretty extraordinary. So what do we find under these climate change induced temperatures? Well, this was a starkly different result. You can already notice that the coral has already started to shut down its growth responses. You see that at day 2, when the tips were injured, we expect there to be no tissue or skeleton growth. But what's really striking here is that by day 12, you have the tip starting to initiate, but it doesn't complete. It shuts down. There's not enough resources to actually enable the coral to continue to provide enough nutrients to build its tissue and build its skeleton. Now, when we talk about the GFPs again, you can see that the corals actually shut down that response. You saw in the last image, I'll just go back one, that it's highly fluorescing. It's bright on these top parts of the coral, but you notice that it's actually the opposite now at 32 degrees. It's actually shut down the production of GFPs, and the coral therefore is not protected against UV radiation and other external physical damages, and therefore cannot protect the newly growing tissue and skeleton from growing to its full extent. And of course, the SEM images are reflecting the same. The skeleton starts to build. You can see a bit of it come out at day 10, but then it shuts off at day 12 and it ceases to grow. And therefore, it shows that even under the bleaching temperatures, these corals are at the extreme limits. They're not going to be able to recover to their full extent. And that is incredibly worrying here if we see incremental increases in sea surface temperatures going forward into the 21st century. So once again, the corals in these hot tanks do not grow back. And that is a stark reminder of the impacts that climate change is going to pose to these very fragile branching coral species. So what is this impact that I'm going to talk about here? So firstly, from our results, are we likely to see reef recovery under these conditions? So in the case of a 32-degree increase in temperature, we're likely to see these branching coral dominated reefs transition into these very degraded, fragmented reefs, where we're going to be left with only the hardy corals, our mounding corals, our encrusting corals that are left to actually nourish that reef system. So if we do not act now, we will most likely see these very complex reef structures transition into very homogeneous, flat, barren reefs with very little productivity. And just thinking, if you look on the left-hand side, you see an abundance of beautiful small reef fishes hanging out above their coral colony. These are coral-associated reef fishes, which rely on the colony as habitat. We're going to lose a large chunk of that biodiversity if we lose those more fragile coriombros, tabular, and branching corals from these habitats. And once again, we'll end up with these very degraded reefs, which is a stark reminder from a lot of places in the Indo-Pacific, where reefs have already transitioned to these states and are unable to rebound and recover to these more complex structures. However, there is always a way out of these. So going forward, it's going to be really important that conservation efforts are actually targeting regions where you have a large diversity of coral species, not just branching corals, but also one of those more complex, slow-growing coral species, because it's going to be those reefs that are going to actually be the future reefs of tomorrow. Now, with increased CO2 mitigation, so reducing our emissions, this will enable the reversal of some of these more detrimental impacts on these reefs. However, if we do not see any of these stark measures coming into effect over the next decade, reefs, complex reefs, which have these very fast-growing coral types, will be lost eventually, and we'll be left with very simplistic reef structures. So going forward, it's going to be a means of preserving reefs which are high in productivity, high in coral diversity, but also being able to very structurally target which regions are going to be the future reefs. We cannot save every reef on the planet. It's going to be looking at which reefs have these larger benefits, both in conserving biodiversity and how those reefs will broader support other processes, like in my case, the building of reef islands going forward. But We can all do our part by being more conscious about our energy usage, and hopefully into the future, we can then start to transition to much more renewable technologies, renewable sources of energy, which are already in motion, to help better safeguard these reefs under the impacts of climate change, and then better, as a result, conserve their complex biodiversity. So I'm not going to go on any further. I thought there's a lot of information there from that experiment. So please fire away with any questions that you have with regards to this. And I hope you enjoyed it. So I'm happy to take questions. And thanks for listening. And I hope you got a bit of a perspective of how you can use a really diverse skill set over such a short career time frame.
Thank you so much, Josh. Can we give a round of applause? Beautiful. So I've got a student here. She's asking, so a lot of the research says that bleaching events happen between a change between 1 and 2 degrees, but yet this is showing like a 10-degree change. So she's just asking about the susceptibility of bleaching and the thresholds of temperatures.
Absolutely. So she's absolutely right. Bleaching actually happens between a degree change, as mentioned. So Acropora aspera, if you think about the thermal tolerance of a given coral species, it's not just going to flourish in the range of 26 to 24 degrees. It's actually going to have a large extent of range that's going to cope with temperature. But we've just been able to target that it's probably over 30 degrees now that aspera can no longer cope with high temperatures. So most corals have a window of thermal tolerance, as I'm going to call it. So it's that upper threshold above 30 degrees. So between, in this case, I'm going to guess between 1 to 3 degrees for Acropora aspera. It's pretty much dictating whether it bleaches or not and how susceptible it is over that upper part of its thermal tolerance. So at the upper part above 30 degrees, it's very fragile. But of course, below 30, it's going to have a extensive amount of resilience to temperature because it's actually one of the hardiest branching coral species in those environments. So absolutely right. They do have a bleaching window about 1 to 2 degrees. But remember, that's just at the top of the threshold. It doesn't include the entire tolerance of the species that I've covered in this experiment. But that's a really great question. That's an awesome question. Very technical.
Perfect. Have you had a look at playing around at all with any type of branching coral that are more tolerant to temperature change, and in terms of sort of, I guess, artificially creating complex reef structures?
Absolutely. So not me per se, but I have colleagues who are working in that space. We tend to use these reef flat specific corals as really good test subjects because they already are in a very how can I describe it? They're in a very extreme environment compared to the rest of the reef. So aspera is one such example, but we also have other species such as Millipora, which exhibit a large range of habitats. They occur in these very shallow reef environments, such as the reef flats, which are periodically exposed almost at low tide. And it's these species that are actually being targeted to understand these better because these are the most resilient species to stress. So aspera is probably up there with one of those resilient species, but it's not the most resilient to temperature stress. I think other studies have targeted Acropora millipora and other branching types such as Pasillipora, which have a bit more higher tolerance to those temperature stresses. But it's always going to come down to which environments they're sampled from. So you want to be sampling corals which are actually already in extreme conditions. So for example, a colleague of mine who was at UWA a couple of years ago, she worked on Acropora aspera in the Kimberley on the intertidal reefs. And these reefs are they're extremely exposed at low tide for over 3 to 4 hours, and therefore they're really under heat stress. But they've actually adapted over time to those conditions. So it's the same species as the one that I've focused on, but in an entirely different system. So it's once again, it's actually system dependent. So, but once again, she's targeted a very resilient species, which was Acropora aspera in this case, but from the Kimberley.
Amazing. I think we've got one more question. Amy's going to come up. She's going to ask a question.
So is the tolerance of coral reefs in Western Australia different to like the Great Barrier Reef?
That's a great question. So once again, yeah, it actually would be definitely, in my opinion, more resilient and much more tolerant, particularly because of our reefs in Western Australia are far more remote than the Great Barrier Reef. So for example, we have, you know, most of our reef systems concentrated from the Abrolhos up to the northwest shelf of Western Australia. We do have a lot of flushing from a longshore transport of currents down the coast, which actually helps to cool the reefs a bit better than the Great Barrier Reef. But we don't have as much like nutrient pollution, such as eutrophication, entering our reef systems, which could be causing bleaching as well. So in terms of how remote our reefs are, that's kind of been the saving grace to why Western Australian reefs are a bit more resilient than our GBR counterparts. And they're going to probably be the most well-preserved reefs, I think, if the GBR was to become more degraded into the future. So we're quite lucky here that our reefs, being as isolated as they are and not having as much coastal development, we have very few towns along northwestern Australia. That's actually helping to safeguard these reefs a bit more. And not to mention some of our offshore reef environments like Scott Reef, the Rolly Shoals, they're very far offshore, so that they're not going to actually be experiencing a lot of these coastal effects. And being in more oceanic environments, they tend to be a bit more kind of buffered from these high energy, from these high temperatures. Yet again, they're more exposed to, say, cyclone severity in some regions in that area of WA as well. So it's, in general, we've definitely got much more tolerant reef systems, in my opinion, in Western Australia compared to the Great Barrier Reef.